Hi, everyone. Welcome to another edition of Lunch with a Scientist. I'm Leah with Headwater Science Institute, and today I'm very pleased to introduce you to Naomi Atkin, who's a friend of Headwaters in a variety of ways, which you'll hear about later. Naomi earned a BS in genetics from UC Irvine in 2015 and defended her PhD thesis in April 2021 and just received her PhD. So big news in her world. And her work in the Wang Lab focuses on understanding the role of DNA breakage in cancer susceptibility. Specifically, she has worked on mapping DNA breaks using samples from both AML and breast cancer patients in order to identify break-prone regions of the genome that can then be used to create a DNA fragility and cancer susceptibility test. So if that sounds like a mouthful, don't worry, we'll hear all about it today. And this work has led to the development of a break mapping method that is compatible with extremely low amounts of input material. Outside of the lab, Naomi enjoys reading, volunteering as head copy editor at the Journal of Emerging Investigators, which Headwaters students frequently submit to. She also enjoys baking cookies. So we're super excited to welcome Naomi Atkin.
Hi.
Hi, how are you?
I'm well, thank you.
Great. Well, congratulations on getting your PhD, a huge accomplishment.
Thanks.
And we're super excited to hear what your research was about. So I'll bring your presentation in and I'll let you take it away.
All right. Okay. So, like Leah said, I've recently defended my PhD thesis at the University of Virginia. And over the last 6 years, my research has focused on understanding what factors like proteins or chemicals that contribute to DNA breakage in the genome. And really, this is important to think about and study just because DNA breaks actually happen relatively often as normal processes in the cells. But if they aren't repaired well or if factors increase DNA break frequency, then there can be bad consequences like DNA mutations, which could then put people at an increased risk for cancer. So today, I'm going to be talking about how DNA breakage at key regions can be used to predict cancer susceptibility. And kind of like I hinted at before, if we can better understand this preferential breakage, as well as what mechanisms are contributing to it, we can use that information to create tests that better measure and predict cancer susceptibility before cancer has formed, which ultimately is going to help us improve patient outcomes. So very briefly, a majority of cancer-driving genes are subject to somatic or non-hereditary mutations. And 65% of those cancer-driving genes have the potential to be involved in chromosomal abnormalities like translocations, amplifications, and deletions. And what's really important about this is that a DNA break must precede the generation or formation of these chromosomal abnormalities. Therefore, we hypothesize that DNA breaks at key regions can be used to predict cancer susceptibility. And that's because if a region has more DNA breaks, it has more opportunities to lead to these chromosomal abnormalities. And really, the reason we are interested in this is because currently cancer screening methods heavily rely upon physical or genetic abnormalities like, say, tumors or the presence of these mutations to already be there to be detected. And what this means is that at that point, if someone already has a tumor or has one of these mutations, obviously the cancer is progressed and survival outcomes or just general patient welfare are not as good as they could be. So we propose that measuring DNA breaks can help us identify people who are at risk for cancer before these mutations happen and before cancer happens. But in order to be able to do this, we needed to develop a method that would allow us to sensitively identify and quantify DNA breaks, and was compatible with low amounts of starting material, since we don't tend to get a lot of material from patient samples for various reasons. And so I know this is kind of unfamiliar for a lot of people, but there are a few methods out there that people have used to directly map DNA double-strand breaks, or DSBs. And really the one common thing that I want you to focus on with these is that they all require a lot of cells in order to do the break mapping. And we think this is most likely due to inefficient reactions during the break mapping process, since most of the reactions during these various methods happen within the cells or within the nuclei of the cells, which means that all of those reagents and molecules and chemicals have to pass through cellular and nuclear membranes in order for the reactions to occur. So, as I mentioned, we want to be able to map DNA breaks in patient samples. So using 5 to 70 million cells is just not possible, because we don't get that many cells from patient samples. So therefore, we decided to try and use one of these methods and adapt it so that we can use purified genomic DNA as a starting material, because we think that will help improve reaction efficiency and will help us, in the end, use a lot less material to map DNA breaks. So in that line of thinking, we actually chose to adapt this method called DSP capture because it was the most similar to a previous gene-specific break mapping method that we had in the lab, and that was because it directly mapped DNA breaks with sequential or stepwise adapter ligation, which I will explain on the next slide. And compared to the other methods shown here, it tended to have better signal to background ratio, which will allow us to identify break-prone regions much better. So overall, we felt it was the best option for adapting a protocol to have one that uses low amounts of genomic DNA as the starting material, and which is going to make it much more compatible with patient samples. So this slide will maybe end up looking a little complicated, but I will explain what the important parts are of our genome-wide break detection and sequencing method. So really what this amounts to is in the cells and in the DNA, there are DNA double-strand breaks, which are breaks that span both strands of the DNA. And we can see in red, we're indicating the single nucleotide position of the break that happened within the cells naturally. So what we do is, after we've carefully isolated our genomic DNA to make sure we don't introduce artificial DNA breaks, we repair the ends so that now they are blunt, so the two ends are even, but still maintaining the integrity of that nucleotide position of the break. We then attach what we call an adapter to that end, which this is going to now protect that break end while also marking exactly the position of the break that occurred within the cells. Then we do some downstream steps where we then break up so we have smaller fragments, and then we do another round of end repair and a final ligation so that these fragments can now be sequenced using Illumina or next generation sequencing. So we get a lot of data from this. So we get gigabytes of data per sample. So we developed a pipeline that will allow us to analyze the data from every sample that we're getting. But really the important takeaway here is that we focus primarily on this little first nucleotide of read 1, which mapped that break that happened within the cells. And this allows us to very sensitively identify the position of any break that we identify. Okay. So the thing that I've mentioned that is so important to what we do with using patient samples is our variations or our adaptations that we've created with this method. And so we have the original one that maps DNA breaks in nuclei, and we've already brought down the amount of input required for this due to increasing our efficiency of our reactions. But most importantly is this purified genomic DNA adaptation that we've got. And with this, we've been able to achieve a 60 to 850-fold reduction of input material that we use, which has then allowed us to be able to map DNA breaks in patient cells. So, okay, and then the main thing here also is that during this whole process, and when we isolate our genomic DNA, like I kind of hinted at earlier, we need to make sure that we're not introducing significant amounts of outside breaks or ones that we've generated. And so we have a couple steps within here that we do to make sure that this isn't happening, because if this were to happen, we would then have a lot more background into our data that we've generated, which would make it very difficult to identify true break signals that we're interested in identifying. Okay, so now that we've got this method that is going to help us generate reliable and sensitive data, we could then ask, well, what features in the genome are contributing to DNA breakage? Now, previous studies have shown that this enzyme called TOP2, which cuts both strands of the DNA, if the step that the enzyme is capable of doing, which is to join the broken ends back together, if that religation step fails, whether, just inefficiency of the enzyme or chemicals that inhibit or poison TOP2, so if that religation step fails due to those factors, the DNA breaks can persist if not repaired. Additionally, there are sequences in the genome that can form these things called alternative DNA secondary structures, like this DNA hairpin. And generally, these are just alternate conformations that the DNA can take that is not a typical DNA helix. So what's important to know about these structures is that regions of the genome that have the potential to form these alternative DNA secondary structures have been shown to be enriched with DNA breaks. And more interestingly, when we take sequences that have the potential to form these structures and insert them into non-break prone regions of the yeast genome, we can see a significant increase in chromosomal rearrangements or moving of large chunks of chromosomes around the genome. And finally, we're also interested in looking at these chromatin organizing proteins, CTCF and cohesin. And that's because the CTCF and cohesin binding sites have been shown to be enriched for TOP2-induced DNA double-strand breaks. Now, other studies have previously focused on how these features independently contributed to DNA breakage, but we explored how they function together to directly contribute to DNA breakage. And obviously, since our end goal is to understand and use information on DSBs to measure and determine cancer susceptibility, our next question is, are these features relevant to cancer? Short answer is yes, but more detailed is that TOP2 poisons, or those chemicals I mentioned that inhibit TOP2 activity and religation, treatment with these during chemotherapy is actually directly associated with an increased risk for developing a therapy-related acute myeloid leukemia, or AML, which is just a AML that develops after you've been treated for a primary cancer like breast cancer. And additionally, the oncogenes, or cancer-driving genes called RET and MLL, they're breakpoint cluster regions, so regions within those genes that are more prone to breakage and being involved in translocations. Those breakpoint cluster regions are also TOP2 cleavage sites. And regions of the genome that have the potential to form alternative DNA secondary structure are enriched for oncogenes and tumor suppressors. And finally, CTCF and cohesin binding sites are frequently mutated in cancers, and if cancers have these binding sites mutated or mutations of the proteins themselves, those cancers tend to be more aggressive and have a worse prognosis for the patients. And finally, what was interesting to us, because we do focus on AML, is that 12 to 20% of AML or myelodysplastic syndrome have a cohesin mutation. So therefore, these mutations are relevant to cancer and appear to be good candidates to consider when assessing DNA breakage and cancer risk. So now that we know that these features that are kind of interesting for our lab are independently associated with DNA breakage and cancer, can we then use this information to better help us understand and determine cancer susceptibility? And more specifically, will DSBs at key regions at key regions defined by these features be able to predict cancer susceptibility, which again will help us better identify and predict who might be at risk for cancer before they get cancer. Now, because we want to create a DNA test for cancer susceptibility, we needed to make sure that we can actually see differences in DNA breakage in patient samples, because prior to this, no one has been able to map DNA breaks down to that nucleotide single position level across the whole genome before, because none of the methods were compatible with the amount of material that there is with patient samples. So to do this, we set up a proof of principle experiment in which we already know the mutation and the rearrangement or translocation status of the patients that we tested. So here we compared the DNA breakage in AML fusion genes, specifically the MLL fusion gene, in non-fusion control AML patients, so those whose AML was driven by a point mutation or a single nucleotide change of a gene, as well as MLL rearranged remission AML patients, so those with an MLL fusion or translocation. Now, we do this because we know the remission patients were at one point susceptible to a rearrangement-driven cancer. And therefore, we hypothesized that they must have been more prone to DNA breaks within the gene that caused their fusion, which in this case will be MLL. And meanwhile, we predict that the non-fusion patients will not exhibit this preferential breakage in MLL. So, and really it's important that we used remission patients for our MLL rearranged patients, because the original rearrangement is no longer present since they're in remission. And so that means that we're then measuring the inherent, or like what is specific to their normal cells, the inherent break susceptibility of their normal cells, and not that of their cancer cells. So to map DSBs in patients, we've come up with this protocol where we isolate peripheral blood mononuclear cells, or PBMCs, from leukemia patients using FICOL. Now, really, what you can think about FICOL is it's just like a density gradient, where you put blood on top of this FICOL, and we run it through a centrifuge, which just spins the tube really, really fast. And what happens is the denser red blood cells go to the bottom, and the lighter plasma and PBMCs go to the top. And we can just simply carefully take off that plasma, and then we can access those PBMCs and do whatever we want to do with them, which in this case, what we do is we grow them for a little bit, and then we can treat them with chemotherapy, chemicals, and topoisomerase 2 inhibitors like etoposide, which I mentioned earlier. So this is going to allow us to enhance any subtle DNA break tendencies or differences that might not show up until stressors like chemicals are present. And this is going to then allow us to more sensitively assess cancer susceptibility. So using our low input break mapping method, we carried out that proof of principle experiment that I explained a couple slides ago. And in this case, we looked at DNA breakage in our non-fusion patients and in MLL rearranged remission patients. And here we're just looking at their DNA breaks at that specific MLL gene and its breakpoint cluster region. So the region that is most prone to breaks in other patients, as well as the region that is involved in the MLL translocation. So you can see here just by eye that the MLL rearranged remission patients appear to have a little bit more DNA breaks in this region compared to the non-fusions. And when we quantify that those DNA breaks in that region, we can see indeed that there are significantly more DNA breaks in our 3 rearranged patients compared to our non-fusion patients. And interestingly, when we look at another leukemia-associated translocation gene called RUNX1, we don't see that trend anymore, even both by eye and when we quantify our data. We see no significant difference between our non-fusion patients and our MLL rearranged remission patients. And also kind of interestingly for us is that the DNA breakage level that we observe at RUNX1 isn't as high, even just baseline, compared to the MLL region, which is indicating to us that one, MLL patients are more break prone within the MLL region, and that this is specific to the MLL region and not just another translocation gene. So then what we did next is we decided to treat our patient samples with that TOP2 inhibitor called etoposide. And importantly, we treated with doses that mimic what is left in the blood after chemotherapy, but they don't cause cell death. So what we're seeing is not just because the cells are dying. The cells are still alive, they're still happy. So when we did that, we can see that in our MLL patients in blue, we see a significant increase of DNA breaks after our treatment with etoposide. And we don't see that trend in the non-fusion patients. And then again, importantly, we don't see any significant difference between our non-fusion and MLL patients, nor do we see any significant differences following etoposide treatment in either patient group in RUNX1. So altogether, what this is showing to us is that the MLL rearranged remission patients were preferentially susceptible to breaks at that MLL breakpoint cluster region. And this was specific to MLL and not other fusion genes, which means that we can, or that mapping DNA breaks is capable of differentiating between a susceptible, our MLL rearranged remission patients, and a non-susceptible, our non-fusion patient groups. So because we have this genome-wide data, we then wanted to identify regions with equal or greater breakage than that observed at the MLL region so that we can better understand the mechanism that's driving this differential fragility that we observe in our 2 patient groups. And with it, we want to do this because we hope that it's going to help us work towards building a fragility and susceptibility test in the future. So with help from another lab here at UVA, we've done this unbiased analysis, which just means we didn't go in with any preconceptions about what we expected to find. And what they did for us is they took our patient data and they looked for transcription factor binding sites at which our MLL patients had more DSBs than our non-fusion patients. And when they did that, they actually found that the top hits, so the sites that had the biggest difference between our MLL patients and our non-fusion patients, our top hits were the cohesin complex subunits RAD21 and SMC3, as well as CTCF, which was interesting to us because that is matching a little bit of what's in the literature about what contributes to DNA breaks. So it was a nice, good confirmation on that front. So based on this, as well as the fact that the MLL breakpoint cluster region is also a TOP2 cleavage site and a CTCF binding site, we used publicly available data sets to identify which sites were specifically preferentially break prone in our MLL samples. So we did a couple stringent threshold and filter steps, which you don't need to be concerned about, which then gave us about 200 to 500 protein binding sites that demonstrated breakage that was equal to or greater than that was observed in our MLL region on the previous slide. And just so you can get a sense of what kind of differential breakage we're observing, this is just the DNA breaks at each protein binding site for our 3 MLL patients in blue and our 3 non-fusion patients in orange. And really what I want you to take home from this is that there's a very noticeable difference in the amount of DNA breaks at our sites that we've identified between our non-fusion and our MLL patients. So indeed, our MLL patients are breaking more at these regions than the non-fusion patients. So while this information is informative, we still wanted to narrow down these sites so that we can use them to evaluate susceptibility in patients. And so therefore, we next identified sites that were common among TOP2, CTCF, RAD21, and SMC3. So with that, we identified 34 common regions, and we found that when we calculated the cumulative breaks within our MLL patients and our non-fusion patients, that there was a significant difference between the cumulative breaks observed in our 34 common regions in our MLL patients compared to our non-fusion patients. And interestingly, we observed a greater difference when we assessed breakage at 34 regions than we did at just that one MLL region, which is indicating that measuring DNA breaks at multiple regions is going to give us a better assessment of DNA fragility in patients. So interestingly, when we looked at how these regions responded to that TOP2 inhibitor etoposide, we found that in our MLL patients, compared to untreated, the MLL patients had significantly more DNA breaks after treatment with etoposide. But when we decided to look at this a little more closely, we looked at these regions one by one and how they responded to etoposide. We actually found that 10 of the 34 regions did not respond to etoposide. So that means they didn't show any increase of DNA breaks from untreated to our etoposide treatment. So we wanted to see what was going on with those. So we looked at the DNA breakage values at those 10 regions, shown here as the little red dots, and we compared those to the genome-wide DNA breaks for each MLL patients. And so you can see here that very clearly those 10 regions are greater than the general distribution of the DNA breakage genome-wide for each patient, which is indicating to us that these 10 regions were just inherently break prone, and that it's possible that etoposide couldn't have an additive effect because they were already breaking so much compared to, our baseline breakage level. So overall, what this is telling us is that there are 2 classes of sites within the 34 common regions that we identified. One that is inherently break prone, so these 10 regions, and one that actually is sensitive to TOP2 or etoposide treatment. And both of these classes are important to consider when trying to evaluate our risk prediction. So based on these findings with the AML patients, we then wanted to determine whether we could identify individuals at higher risk for cancer without knowing their mutation status. So to do that, we considered the fact that treatment with TOP2 poisons or inhibitors during chemotherapy is directly associated with an increased risk of developing therapy-related AML or TAML. And we also considered the fact that due to how they are treated and what they're treated with chemical-wise, breast cancer patients are most at risk for developing therapy-related AML. Therefore, we used whole blood from breast cancer patients, and we isolated the PBMCs, just like I described a few slides back. And importantly, we only used patients that were pre-chemotherapy. So they haven't received chemotherapy yet. These should be relatively, quote unquote, normal samples that we're getting. And the important fact is that we only included patients that would eventually go on to receive DNA damaging agents like TOP2 inhibitors, which includes etoposide and this other TOP2 inhibitor, doxorubicin. So I know this slide is probably a little chaotic, but what I want you to focus on is when we measured DNA breaks in our 6 pre-therapy breast cancer patients in 3 TAML associated regions, so MLL, CBFB, and RUNX1, we can see that the MLL region is by and large demonstrating more DNA breaks even in the untreated samples compared to both CBFB and RUNX1, which is telling us that the MLL region is just more sensitive to DNA breaks compared to the other 2 regions. Now, when we look at this a little more differently for the MLL region, and we look at each individual breast cancer patient, we can see actually that 4 of our patients, breast cancer 1, 2, 5, and 6, don't really show a strong effect when they're treated with our TOP2 inhibitors, etoposide, and doxorubicin. But when we look at breast cancer patient 3, we can see that actually they start in their untreated sample very high, and then as a result don't really show a response to chemical treatment. But when we look at breast cancer 4, who doesn't show any DNA breaks in this MLL region, they then show a very strong increase of DNA breaks after both etoposide and doxorubicin treatment, indicating that this patient is most likely more break prone in this region compared to the other patients. And interestingly, these results where we've identified 1 patient out of our 6, or 16% of our patients, as potentially susceptible to an MLL gene fusion event, this rate actually matches or almost matches the frequencies for these TAML associated fusions that are in other studies. And specifically, other studies have shown that about 15% of TAML cases are driven by MLL fusions, and about 8 and 5% of TAML cases are driven by CBFB and RUNX1 respectively. So this was kind of interesting to us. We didn't expect to actually identify someone who might be preferentially susceptible to chemical-induced breaks with such a low number of patients, but it was interesting to us to identify this. So because we've found with those AML patients that assessing DNA breaks at more regions could actually provide us with more power when predicting susceptibility, we next looked at DNA breakage with these 6 patients at those 34 common regions that we had identified earlier. So with this, we found that at our MLL susceptibility sites, or those 34 common regions, we can confirm that breast cancer patient 3, shown in peach, is intrinsically more break prone, as shown by their untreated sample being above all the other 5 patients. And then when we looked at breast cancer patient 4, shown in red, we can see that indeed, compared to the other 5 patients, they do show more etoposide-induced DNA breaks at these sites. So this is further supporting that these MLL susceptibility sites, or those 34 common sites, have the ability to predict both intrinsic and treatment-related risk. And overall, these preliminary results that we've got are indicating that mapping DNA breaks in key regions can predict cancer susceptibility, but with the caveat that follow-up on this breast cancer patient 4 is needed to actually confirm that this patient is more at risk for therapy-related AML. So in conclusion, CTCF, TOP2, and alternative DNA secondary structure are directly contributing to DNA breaks in cancer patients, as demonstrated by our proof of principle experiments that showed the MLL region can be a predictor for MLL rearranged AML, in that TOP2, CTCF, and cohesin sites also exhibit preferential break sensitivity in susceptible individuals. So therefore, DSBs at key regions can be used as a tool to predict cancer susceptibility in patients. So in the future, our work, we're hoping to kind of delve a little deeper into what mechanisms are contributing to chemical-induced breaks, because we think that there might be a damage type or a chemical-specific dependent effect on DNA breaks and susceptible regions. And as the last couple of slides indicated, we want to use this to help predict therapy-related susceptibility by assessing DNA breakage in pre-therapy cancer patients after our stress test. And that's just going to include looking at more breast cancer patients, maybe looking at other cancer patients. And finally, overall, we want to use our approach of mapping DNA breaks in patients to help get a better understanding of risk assessment for other cancers, like solid cancers, like breast cancer. So with that, I would just like to use this last little slide to acknowledge everyone that has helped contribute to this and other projects over the last 6 years.
Great. Thank you very much, Naomi. That was awesome. I thought it was really fascinating that you talked a lot about something that impacts a lot of us. Cancer is unfortunately on the rise. So I'm really excited to dig into some questions. But first, I just want to add on that at the beginning of the talk, we mentioned that Naomi is also involved in the Journal of Emerging Investigators. And that's a journal that we have our students submit to when they're done with their own research projects. So really cool full circle connection here. And our other fun surprise is we've actually got Alec Atkin here. Hi, Alec.
Hi, Leah.
And so Alec is our board of directors treasurer, and Naomi is his niece. And so we're very excited to have this friendly connection as well. So we are going to dig into some questions, but Alec is also going to help me ask those questions today.
Great.
So Alec, why don't you take it away?
So first off, congratulations on getting your PhD. I know your family is really excited about that. It's a really big accomplishment. So I was listening to the talk, and I have just a few questions that are related to maybe vocabulary. Mm-hmm. So what is genomic DNA, and where is it found? Is that just like cells, or is it something special?
So genomic DNA is just the DNA that is in the nucleus of the cell. I just use that to distinguish between the other break mapping methods that keep the cell intact when they map the DNA breaks. So everything that is needed to map the DNA breaks has to go through both the cellular membrane and then the nuclear membrane. Whereas what we do is we very, very gently pop open the cells and then very gently pop open the nucleus. So that way we can keep our genomic DNA as intact as possible. So that then all of the things that are needed to get the reactions going don't have to go through multiple barriers to reach the DNA, can directly access the DNA.
Okay, so basically you're just taking the DNA out of the cell really carefully so that you can use it, and throwing away all that other stuff. Okay. I was interested in when you have double-stranded breaks, DSBs as you call them, and they form alternative DNA secondary structures. Okay.
Yes.
So how many different structures have you seen?
Technically, okay, I'd have to count them off because I have to picture our diagram in my head. So there's the hairpin structure, which I showed on my slide. There's actually a cruciform structure, which is kind of like if you think of multiple hairpins, so you've got top and bottom and side. So a little bit of a clover shape a bit. And then there's these are probably very technical. There's G quadruplexes, which are just like rings of G's stacked on top of each other. So it forms like kind of like a block. And then you can actually have different twists to the DNA. So the DNA not always, but the typical form the DNA is in cells is B form DNA, which just means it has a certain type of twist. So you can have Z DNA, you can have A DNA, I think is another one. There's just a whole bunch of different conformations the DNA can take based off of the conditions it's in, or whether it's separated the strands and now it can like form back onto itself.
So what happens? I mean, does that DNA actually do anything? Like we say, all of our DNA will cause some sort of feature within us. So if you have a different shape, does it just kind of exist with no purpose, or does it end up actually doing something?
Some of the shapes actually do have a purpose. So off the top of my head, I know that so transcription start sites, which are the regions that start, or where the start signal is for that process where we go from taking the DNA to the RNA level, which is transcription. A lot of those are actually enriched for alternative DNA secondary structures, and people think that those structures are helping to promote transcription.
That's interesting.
Yeah, and on the other end, structures can impede things. So during replication, which is making more DNA, if there's a structure, downstream somewhere on the DNA, those proteins that are carrying out replication, if they hit them, it can actually stall or stop replication and cause the proteins to fall off. So they can do both. They can inhibit and, help.
Okay. That's interesting. I wondered about that. Mm-hmm. And it seems kind of a real fascinating part, 'cause I had never thought that DNA would break and reform into different structures.
Oh yeah, the DNA does a lot more than I think most people realize in terms of like just how dynamic it is.
I like those naughty kids at night getting into trouble. Okay, I have a big question here for you.
Uh-huh.
Do you think scientists will ever find a way to stop cancer from ever occurring? 90% of the time. I won't say 100%.
No, unfortunately. I mean, it's as it's becoming known, it's just like it's a disease of aging. So the longer you live, the more likely you are, unfortunately, to get cancer, just because you've now lived longer, you've had more time for mutations to kind of slip through the DNA repair process and to stay and be bad ones potentially. I don't know that you can necessarily cure it, which is why we're focused on early detection, because for a lot of cancers, the early detection really isn't ideal. Like I said, you have to have a tumor present, at which case, depending on when you find the tumor, you're either worse or even more worse off. And so we think that with early detection, at the very least, you have the ability to catch it sooner, so you have more options for treatment, treatments that maybe aren't necessarily as hard on the body. And at that point, if you catch it earlier, also the cancer has less mutations that it's acquired during its cancer process. Right. So then you're better. It's a simpler type of situation to try and attack and target. You're not battling as many demons and having it mutate even more and gaining resistance to therapies. So,
okay.
So I have a question that kind of tags off of that, Naomi.
Mm-hmm.
Most of the scientists that we meet are doing incredible work on their own, but they're also looking at research that can inform what other scientists can do as well. So now that we know some of the potential maybe precursors for cancer, or how to better identify who may be susceptible, how does this trickle into other areas of science or the medical field where we can actually apply this to treating or looking at patients?
Oh, goodness.
Big question, huh?
I know, because a lot of what I've done obviously isn't immediately going to be used by any doctor. And especially because I studied one cancer, which is leukemia. And we're pretty comfortable in saying that even within a leukemia, ones that are driven by other fusions, like I mentioned, RUNX1 is another fusion that could happen. We think each individual cancer type or cancer subtype is going to have its own set of like common regions or break-prone regions that can help us measure susceptibility. So in that case, like we're going to have to test a whole bunch of patients in order to get a better idea of what actually is important for each cancer subtype. So that's years and years and years of work that's still to do. And I mean, like outside of cancer, a lot of these principles can actually apply to things that you wouldn't expect. We actually have someone who was one of my like advisors or on my committee. Her lab studies plasmodium that leads to, I believe, malaria. And the mutation process for those plasmodia also, they think, involves the formation of DNA secondary structure, and that being the area that has a DNA break, which is then allowing the plasmodia to amplify the gene that's causing the bad phenotype, or the thing that causes the malaria and makes them resistant to treatment. So like these concepts aren't just specific to human cells either, we don't think.
Interesting.
It's really interesting. Yeah. It seems like there's just so much to it seems like just studying DNA is just so fast, and we're only at the beginning of it. Yeah.
That's exactly why I started in it.
And I'm curious, Naomi, about how you got started. You worked your way to a fairly specific kind of corner of research, but how did you become interested in the field, and what educational path did you take to get here?
Okay. So, I mean, pretty early on, not pretty early on, about halfway through high school, I realized that science is just what made sense in my brain, much more so than other topics. And then I had a really good and enthusiastic AP biology teacher my last year. And I was also taking other science classes, like I think human biology, and then one that was focused on introducing us to different career paths as well. But because of the AP biology class, I realized that DNA was the thing that really got me going. And I was kind of in the back of my mind always aware of like genetic diseases. And so, but it was that class that really made everything click together. So then I looked for colleges that had a genetics major as an undergrad degree, which is not common. And that's how I found UC Irvine, because they have a bachelor's in genetics. And also bonus, their freshman housing community is called Middle Earth. And that really appealed to me. So I went to UC Irvine, and then kind of sort of on a whim, but also a very calculated whim, where I heavily researched all the classes that would be available to make sure I was still graduate on time. I decided to do a year abroad in England, and got to take a cancer class there, which was just on the hallmarks of cancer, which is at the time was much newer than it is now, of an idea. And so from that, I realized I really liked cancer as well, because I figured out I liked problems that are very hard to wrap your brain around, and that was DNA and cancer. So I did an internship at a genetic testing company the summer I got back, and was just looking at a lot of how cancer passed through families. And so that made me more aware of the genetic relationship with cancer, which is funny because I do nothing with hereditary cancer anymore. But yeah, and then that made me realize that I wanted to go into understanding what was, leading to people to be more at risk for cancer. And so I just looked for programs that would allow me to bop around labs until I could find one that worked for me. And it was honestly a chance conversation with a student who was older than me in my program while I was imaging something for another lab. who was like, hey, what do you like studying? And I said, these things. I like DNA when it goes wrong and cancer. And so he was like, actually, there's a faculty member who is going to be graduating 2 students very, very soon and would like another student. And their research is exactly weird DNA that causes cancer. And so that's how I ended up where I am.
Wow. It sounds like a lesson of exploration, to try different things, talk to people, and you never know where you're going to go.
Exactly. If I had not been complaining about just like not being as satisfied in the like trial periods I had in the other labs I had been in, and just like the science wasn't exactly right for me, and I was just explaining, all I had in mind was I like DNA when it goes bad, and if it leads to cancer, bonus. And he knew exactly the person, and it was just like right place, right time conversation.
Wow. Thank you so much for being here to share. Thank you, Alec. This is a really cool special session with you here to ask some questions. And I hope you both have a wonderful day.
Thank you, Leah.
Thank you.